Thank you so much, and good morning to you on the West Coast. I deeply apologize for not being able to be there in person, but I'm really happy to present some of our research work that's been funded by CFRI, and I am forever grateful for the CFRI support, because without this, we could not do what we have done here today. So this morning, I'll talk to you a little bit about this CFTR High Expresser cell that we have elucidated to be a new secretory cell type in the intestine, and touch on the relevance to cystic fibrosis gut disease. So in terms of outline, I'll touch a little bit about the discovery of cheese and the early studies that we conducted on the characterization of these cells and CFTR traffic within these cells, and then touch on the more recent single-cell RNA-seq studies and our studies to determine the cell fate of these cheese cells and recent characterization. And finally, a word about the relevance to CF intestinal disease. So first, I'll just talk a little bit about the small intestine, because not everybody studies the gut, and talk a little bit about the cellular architecture. So the small intestine has the epithelium consists of invaginations called crypts, which are cells that dig deep into the epithelium. These cells consist of the stem cells, Paneth cells, and cells that would migrate up into the villi, which are these long fern-like structures, to generate enterocytes. And then you also have enteroendocrine cells and goblet cells interspersed among the different cell types. And when I entered the field in the late 90s, there was strict division of the functions of cells in the crypt as secretory cells and cells on the villi that were ascribed to absorptive functions. Now, cells in the intestine are polarized, which means that they are specific proteins that are present on the apical membrane and specific proteins on the basolateral membrane that allow polarized transport through the cell, and water traverses through the paracellular pathway. And as I mentioned, in the late 90s, crypt cells were identified as the site of fluid secretion single-handedly, and villus cells were only involved in the function of absorption. And the CFTR channel, which is a secretory channel, was accepted to be on the plasma membrane and confined to the crypts. So I just want you to keep these concepts in mind. So when I entered the field, I used very well-validated antibodies against CFTR that were generated in my mentor's lab at the time. And my project was to localize the CFTR protein in the human and rat small intestine. And what we found, these are images from the very first paper that was published, is that in the superficial crypts and on the edges of the villi were these cells with really high levels of CFTR in the apical domain, not just on the membrane, if you can see here, but throughout the cytoplasm of the cell. And here are several of these cells scattered on the edges of the villi in the proximal small intestine. And these are images from the human. We found similar distribution in the rat proximal small intestine. And here you can see these brightly lit green cells along the edges of the villi with really intense levels of CFTR protein throughout the cell. We also identified CFTR in the apical domain in the crypts, which was expected because it's a chloride and bicarbonate secretory channel. But interestingly, we looked later on at the mouse intestine along the length of the intestine, and we did not find these cells in the mouse intestine. I named these cells CFTR high expressor cells by virtue of the fact that they express very high levels of CFTR, because at the time, the only tool we had to be able to identify these cells was antibody staining. The presence of these cells had been previously been recognized by mRNA studies by other investigators. So when we look into, I mentioned about the superficial crypt. So, cells that are secretory are in the crypt. And so we looked more in the crypt and found that these cells were absent in the deep crypts, which is where most of the fluid secretion occurs, but they were present in the superficial crypts, as you can see here at the top of the crypt. And NKCC1, which is a chloride transporter that is very important in many epithelial cells, including the intestine, that regulates chloride entry from the basolateral membrane into the cell to allow CFTR to secrete the chloride from the apical membrane. We found an inverse relationship between NKCC1 and CFTR in the crypt, but in the cheese cells, you can see exaggerated expression for NKCC1. And this was one piece of evidence that supported the notion that these cells were secretory cells. And here you can see at high magnification the inverse relationship of CFTR and NKCC1 in the crypt in those cells. The other thing that we found that these cells were not present throughout the intestine, and these are images from the rat, along the proximal distal axis, you can see that these cells are in the duodenum and in the jejunum in the rat. They're absent in the colon and in the ileum. And the ileum, is a site that's primarily affected in CF. But in the rat, we quantified the location of these cells, and we found that the most of these cells were present in the proximal jejunum in the rat, right? So they were absent in the colon, absent in the ileum, present in the duodenum, jejunum, but most abundant in the proximal jejunum. So one of the questions that was raised when I first discovered these cells was whether these cells were indeed enterocytes or not. And we could not tell by immunofluorescence labeling whether these cells were neuroendocrine cells, because the distribution of these cells resembled that of neuroendocrine cells. Now, these cells consist about 1 to 2% of the total epithelial cell population. And so to determine whether these cells were indeed neuroendocrine cells or enterocytes, I did some double staining for enteroendocrine cell markers when I was a fellow, but did not see any co-localization. So I went to the Netherlands to learn to do cryoimmunoelectron microscopy, which is a technique using antibodies against CFTR to allow the examination at the ultrastructural level to see the structure of the cells and also to localize CFTR at the structural level to confirm where within the cell was CFTR located. And here you can see on the left, one of these cheese cells on the villus of the rat jejunum. These cells are long and they are electron dense, you can see here. The nucleus is locally, basally located, and you can see the cell extends very into the depth of the lamina propria. And in terms of this CFTR labeling, there was a lot of immunogold label on the brush border membrane, but also we identified CFTR on the membrane of many vesicles within the apical cytoplasm of the cell. This was completely new because, remember, at the time, the folks who were dominant in the field were electrophysiologists, and the accepted view was that this CFTR was only on the plasma membrane where it functioned. So this was a new concept, that CFTR was not confined to the plasma membrane, but was also in intracellular vesicles. And here you can see the gold labeling for CFTR on endocytic vesicles and clathrin-coated vesicles, elongated, robust motor fibers are present in the cheese cells with desmosomes. And in the superficial crypt, we identified gold labeling for CFTR in the Chi cells. So these cells were electron dense. The brush border was slightly shorter than the normal enterocyte, lots of endosomes in the cell supporting the role of trafficking, and lots of motor fibers, which also support a role for trafficking of vesicles throughout the cell, perhaps to and from the brush border. We observed that on the cells on the villi, they had non-absorptive properties. Specifically, they lacked expression of absorptive hydrolases, such as sucrase and lactase, as shown here. You can see the gap in the staining. They also lacked absorptive ion transporters, such as NHE3, shown in red, where there's a gap, which is responsible for sodium absorption. They lacked absorption of the chloride absorptive transporter PAT1, shown here in red. There's CFTR, but no PAT1. And these cells also lacked an important myosin, myosin 1A, which we had shown in the mouse villus epithelium is responsible for the trafficking of CFTR into the brush border. But in the rat, these cells do not have any myosin 1A. But they are able to traffic the CFTR into the apical membrane and the brush border upon cyclic AMP activation. So the question was always, if these cells are trafficking the CFTR, how is the CFTR trafficking into the brush border if the cells lack the important myosin that is the motor that moves the vesicles containing CFTR into the brush border? So that remained elusive. So there were limited tools for us to be able to understand and characterize these cells, up until from 2013 to 2021. And in addition, there were other rare populations of cells in the native intestine that we lacked the tools to be able to understand the role of those cells and to characterize them and to understand their functions. And so that limited our ability to do further work on these Chi cells. But in 2021, single-cell RNA-seq was available, and the first paper was published by Hans Clevers's group in looking at the human proximal small intestine using this technique. And Hans identified the Chi cells in the duodenum by single-cell RNA-seq. And what he found was that these cells were highly enriched for bestrophin 4, which is a calcium-activated chloride bicarbonate channel that is present on the basolateral membrane of cells in the colon in particular. He also found that these Chi cells that were enriched in BES4 were also enriched for other genes that were important in cyclic GMP-activated bicarbonate transport and in the regulation of pH. And here's shown some images of the Chi cells co-localized with GUCA2B, which is a gene that encodes for uroguanilin, which is an intestinal hormone that is normally thought to be present in endocrine cells that secrete hormones. But here is a Chi cell enriched in BES4, but also expressing GUCA2B. So that's sort of novel. Since that paper, there have been several studies using single-cell RNA-seq to identify these cells in the human intestine, but in other species. Unlike our previous studies that showed that the Chi cells were absent in mice, similarly, BES4 cells have been identified in a number of species, but not identified in mouse intestine. In the human intestine, BES4 positive cells have been identified throughout the intestine, not just in the small intestine, but also in the colon. But these BES4 positive cells have been shown uniformly to be enriched in bestrophin 4, but also in genes regulating bicarbonate transport and in pH regulation, including otopterin 2, which is a proton channel, and carbonic anhydrase. But the CFTR is confined only to the cells in the small intestine. There have been some attempt to understand the specification of BES4 positive cells in the intestine. And those studies suggest that in the colon, BES4 positive cells fall in the absorptive line. So stem cells give rise to cells that can be activated by Notch. So cells that are Notch on generally go on to be absorptive enterocytes, and Notch off cells generate secretory cell lines and gives rise to goblet cells, enteroendocrine cells, Paneth, and tough cells. That's the current understanding. So the best for positive cells, what we understand today is that they arise from absorptive progenitors in the colon. So how are the Chi cells specified? We really have no idea. So we began some collaborative studies with a colleague of mine in the genetics department at Yale. Now, remember, we showed that these Chi cells are predominantly on a villus and epithelium, which is absorptive, but they behave like secretory cells, although they are on the villi. They are morphologically enterocytes, but they have morphologic features suggestive of a more secretory type cell because the brush border is much shorter, and they lack the usual absorptive markers that are on the brush border membrane in the villus, and they express very high levels of CFTR in the differentiated organoids, enterocytes. So we collected data, and we just published this paper in development, looking at this self-fate of the Chi cells using the rat small intestine as a model. And we've found that there is a secondary Notch signaling involvement in these cells. So here's a schema of what we found in this study, that basically their secretory progenitors are not off, and what is widely accepted is that these secretory progenitors lead to the development of Paneth, goblet cells, enteroendocrine cells, and tough cells. But what we discovered in our work is that there is a bifurcation along the secretory pathway that gives rise to a progenitor that gets reactivated by Notch to lead to a Chi cell. And this progenitor is PROX1, which is a transcription factor, PROX1 positive and Notch2 positive. And this progenitor gives rise to a Chi cell, which is exclusively MICE1 positive, which is a transcription factor. And that so these cells, the Chi cells, are derived from secretory progenitors. And so, as I show here, it involves reactivation of Notch signaling. And I'll show you some of the data around that led to this schematic understanding. And that there are some similarities to what has been found in terms of the cells, the similar cells, the ionocyte in the airway. So in terms of the Notch signaling, this is some data from the paper. You can see what we did in this study here was we treated the rats with DAPT, which is a gamma secretase inhibitor that inhibits the Notch pathway. And when we did that, the number of Chi cells was reduced by 50%. And you can see on the images and also by the quants that the percentage of Chi cells is down by at least 50%, confirming the role of Notch signaling in these cells. And then in terms of the PROX1 MICE1 stuff, we generated organoids using the rat jejunum because these cells are absent in mice. So this was a completely new model that we had to generate to be able to study these cells. And here you can see in the immature organoid, CFTR staining in the apical domain, and in the mature villus resembling organoid, you can see several of these Chi cells popping up with the yellow staining. Similarly, you can see in the differentiated organoid, the MICE1 positive CFTR, which is a marker now of these Chi cells, and the BES4 positive cells in the differentiated organoids. And at the superficial crypt level, you can see the PROX1 Notch2 positive staining and also the Notch2 CFTR staining. And you can see that graphically on the right here. So PROX1 is a transcription factor that is highly enriched in that progenitor that is common to Chi cells, tough cells, and enteroendocrine cells. And MICE1 is the sole transcription factor that's exclusive to Chi cells, but MICE1 is not found in any other intestinal cell type. The BES4 positive colon cells are PROX1 positive, but they are MICE1 negative. They're not detectable in the BES4 positive colon cells. So as I said, that there are some similarities with ionocytes. We looked at to compare ionocytes and cheese. As you know, ionocytes have been shown to be regulated by FOXI1. So we stained rat trachea with FOXI1 and CFTR antibodies, and you can see the FOXI1 enriched in the nucleus of a ionocyte here in the trachea, marked by CFTR. But in the rat jejunum, there is absolutely no FOXI1 in the nucleus, nor did we find FOXI1 staining in any other enterocyte in the intestine. So the ionocyte frequency is similar to cheese. They also express high levels of CFTR, but the transcription factor regulating ionocyte is the FOXI1. And what's been shown recently is that there is a bipotent tuft ionocyte progenitor that gives rise to ionocytes and tuft cells. The gene expression profiles between ionocytes and cheese differ. There is no cyclic GMP machinery in ionocytes, and there is no BEST4 in ionocytes, but ionocytes express Bartin, which is a ion transporter. And the functional studies on ionocytes are showing both evidence of secretory and absorptive functions. It's really not clear. And finally, there's some evidence that sonic hedgehog signaling regulates ionocyte abundance and function. So go back to the Chi cells. These cells are a novel subtype of epithelial cell that is found predominantly in the proximal small intestine, which is more acidic. And our single-cell RNA-seq studies have identified, like in the human, that these Chi cells are enriched for guanylyl cyclase C, which is the receptor in the cyclic GMP pathway. They're enriched for autopterin 2, the proton channel, a specific acid-sensing receptor, GPR4, bestrophin 4, and MICE1. And here you can see by imaging the enrichment of the MICE1 staining in Chi cells, both on the villi and also in the superficial crypt. Now, what about the CF intestine? We know that in the CF proximal small intestine that the pH is lower than normal. And this lower pH in the CF intestine contributes to a lot of the pathophysiology in the CF intestine. The high acidity leads to more thickened mucus because you need alkaline secretions to loosen up the mucus in the gut. It also leads to intestinal obstruction, bacterial overgrowth, gut inflammation, malnutrition, and importantly, a higher risk for GI cancers. So lots of problems due to the low pH in the CF intestine. And so we looked at our single-cell data. And as I said, we found a lot of genes enriched with cyclic GMP-activated bicarbonate transport and pH sensing. And we then looked for antibodies to localize the proteins that were encoded by these genes. And so you can ignore panels A and B. I just showed you that previously. But instead of the myosin 1A that I talked with you about, which is the motor that regulates the trafficking of CFTR in the brush border in the villus epithelium in mice, we found myosin 1B to be enriched in the Chi cells. So that's good because there is a motor in these cells. Bestrophin 4 we found extending on the basolateral membrane into the lamina propria in the basal process, enriching with TUB2, which is we did not find this surprising because these cells traffic robustly. Uroguanilin, the hormone, which is thought to be in neuroendocrine cells, is highly enriched in the cytoplasm of the cheese cells, also in the basal process. And GCC, keep this in mind, this is a receptor for cyclic GMP that should be on the apical membrane, but here it is enriched in the apical cytoplasm and throughout the cytoplasm of cheese cells. And in some sections, we could see BEST4, not just on the membrane, but in the basal part of the cell. And we used our rat organoid model to confirm some of the localization. And you can see in the undifferentiated, which is like a crypt, you don't see the cheese cells, but in the differentiated organoid, you can see many cheese cells, highly enriched for CFTR and mice1 and also bestrophin 4. So we have a model to study these cells. And I'll switch gears now and talk about GLP-1 because it's relevant to these cells. GLP-1 is the peptide released from L cells that respond rapidly to food intake, and these cells communicate via the gut-brain axis. And here's a schematic diagram of an L cell, and you can see that these cells have a brush border that senses contents in the lumen of the intestine. They also have a very long basal process with vesicles that connect with the enteric nervous system to communicate by gut-brain axis rapid transmission. And you can see that diagrammatically how the communication goes on, right? And so these cells are cells that secrete GLP-1 that has a number of functions, including stimulation of insulin secretion, suppression of glucagon, and a retardation of gastric emptying. And these properties have led to successful development of the drugs that you all recognize, Ozempic and the likes, that have been largely successful for type 2 diabetes and weight loss. And so these are a subtype of enteroendocrine cells called neuropod cells. And these neuropod cells have been shown to rapidly respond to luminal conditions in the intestine and communicate with the gut-brain axis, shown here diagrammatically, through these basal processes and this connection through the basal nerve. So we looked in our single-cell data, and we found that indeed, cheese cells were enriched also with a number of neuropod cell genes, including those listed here. And interestingly enough, GCC, which I mentioned before, is enriched is a neuropod cell gene that is also enriched in these cells. These cells are enriched with CHAT, which is a codeine acetyltransferase. It's a neurotransmitter. So all of these genes encode proteins that help in this communication to the gut-brain axis. And so this is important for diseases such as IBS, because successful treatment for IBS involves the use of cyclic GMP CFTR agonists. Linzess is a drug that was FDA approved. It's a synthetic and potent guanylyl cyclase C peptide agonist that is used for the treatment of IBS-C or constipation because of its activity by activating cyclic GMP, as shown here in this diagram, and activating CFTR to increase fluid secretion. The drug Linzess also decreases pain sensitivity through the nervous system, and the mechanism of action was not really known until recently. We contributed to understanding how Linzess works by doing work on the trafficking aspect, and showed that, in fact, Linzess works by activating a kinase, a cyclic GMP-dependent kinase, to traffic CFTR into the membrane, and that's how the drug stimulates fluid secretion. So bear this concept in mind, because I'm going to show you some data about this later. So uroguanilin, as I said, is the hormone encoded by GUGA2B that was widely thought to be expressed in enteroendocrine cells. And this hormone secretes an alkaline secretion in the proximal small intestine where it's acidic, and that's where it's highly enriched. And it was localized in a paper a while ago to cells that look like this. This looks familiar, right? And this cell was thought to be an enteroendocrine cell in the rat jejunum that is responsible for uroguanylin secretion. And this localization, we used the same antibody that was generated by the investigator who conducted this study to localize uroguanylin in our studies. And here you can see what the cell looks like in the rat jejunum. So lately, more recently, intestinal neuropod GCC positive cells have been shown to be very important in regulating visceral pain in IBS. And here are images from that paper. And here you can see GCC positive cells. These cells have been identified in the mouse small intestine and also in the human small intestine. And they communicate with the enteric nervous system. And this is how these cells seem to regulate visceral hypersensitivity. So we labeled for some of these proteins in the cheese, and we found indeed that here you can see through the images labeled by BEST4 positive that these cheese cells are communicating with TUB3, which is a neuronal marker. They enrich with synaptotrophin 3, a neurofilament medium, calcium binding receptor S100A6, the chat, which is present in goblet cells, but also in cheese cells. And here, the GCC is highly enriched in the cheese cells. And you can see the basal process in the cheese cell there, which is very similar to enteroendocrine cells. And you can see by 3D reconstruction imaging, a cheese cell marked by bestrophin 4 in close proximity to TUB3 positive neurons in the native rat jejunum, confirming that in fact these cells are interacting with the ENS and confirming that they are neuropod cells. So what about these cells in terms of what they're doing? We looked at the trafficking of CFTR and GCC under conditions of acidity, because that's important in CF. And here you can see by imaging and quantification that the CFTR is moving under acidic conditions to the apical domain of the cheese cells under acidic conditions. The GCC is also enriched in these cells, but also in other enterocytes in the intestine, but it moves towards the apical domain of the cell, the cheese cell, under acidic conditions. And we have this data is a pre-publication condition. We also looked at the OTOP2 channel under acidic conditions in the cheese cell. And here you can see that under acidic conditions, the OTOP2 is highly upregulated towards the lumen of the cheese cell in the rat jejunum model of low acidity. Other investigators looked at the BEST4 positive cells in the human colon using fluorescent pH-sensitive dyes like rhodo-red, and they found that the BEST4 positive cells had highly enriched rhodo-red uptake under conditions of low pH. So we did similar studies recently in the rat jejunum organoids using the same rhodo-red dye. And what we found is that the cheese cells are maximally taking up protons from the lumen under conditions consistent with CF when the pH is around 5.5, and you can see the image here on the left. So these studies indicate that the cheese cells are taking up protons when the lumen is acidic and putting the protons into the cell. Well, what do we know about cheese and CF? Absolutely nothing. And that's because these cells are not present in mouse. But we did manage to get tissues from a model of delta F508 rat intestine from our colleagues in Australia. And we looked at the RNA for CFTR and GUGA2B. And in the wild type rat, you can see the cheese cells on the villi and in the crypt with high levels of RNA scattered. But the GUGA2B is absolutely preserved in the wild type. In the delta F508 rat, there are markedly reduced levels of RNA, both on the villus and the superficial crypt. But the RNA for GUGA2B is completely preserved in CF. indicating that gluco-2b is not dependent on CFTR. We also looked at the protein for CFTR because nothing is known about that. So in the wild type, you can see these beautiful images on the villi of the cheese cell marked by mice-1 in magenta and lots of CFTR. In the
Dr. Ameen,
yes?
sorry, just a quick time check. We only have a few more minutes. I just wanted to see how much more there was and if
I
we,
have 2 more minutes to talk. And
okay.
so we have markedly reduced CFTR and no CFTR in the knockout, but you can see the mice-1 is positive. The GCC is highly upregulated in the delta F508 condition, and the mice-1 positive cells are present in the knockout. And you can see we found that the Chi abundance increased about threefold in the delta F508 intestine. And here you can see the data on the GCC. And so what we have found from our studies is that Chis are a unique secretory cell type in the proximal small intestine. These cells express very high levels of the hormone uroguanilin and high levels of GCC, but they are enterocytes. They are neuropod cells. They sense the pH in the lumen, and they respond to pH, to the changes in the pH in the lumen by taking up protons under conditions of acidity. And we believe that based on our preliminary studies that they play a significant role in CF pathogenesis in the intestine. So I'll stop here. I just want to thank CFRI for the funding to be able to do the research that we've done. This work was also supported by the NIH and CFF. These are some of the trainees who've worked in the lab and to help generate the data.